So next, really a pleasure to introduce Vanessa Wheeler. Vanessa is a associate professor in the Center for Genomic Medicine at Massachusetts General Hospital, and she's this year's awardee for the Leslie Gehry Brenner Award. And this is an annual award that Frank Gehry had provided to the HDF for innovative research in Huntington's disease on an annual basis in honor of his daughter, Leslie Gehry Brenner. And Vanessa was unanimously, overwhelmingly selected this year to be the prize winner. And her research, we just finished an incredible workshop this weekend on somatic repeat instability, where the repeat can expand into very large numbers in the brain in individuals with Huntington's disease. And Vanessa has, and I'm not overstating this, has really been an incredible leader in this area. It's been her work significantly over the years that has led to our understanding of the mechanisms involved in the proteins that might modulate disease, and also the genetics that is now implicated this process in Huntington's disease and as a potential therapeutic target. So Vanessa, thank you so much and welcome.
Thank you, Leslie, for a very kind introduction. Sorry, my voice is gone. So I'm really grateful to the HDF for this award. Really, I'm truly honored by it, and really it's been a pleasure and an honor to have worked for over 20 years now on HD in this field, and it's a really, really exciting time, I think. And as Leslie said, there was so much energy at this past workshop around somatic instability, and I'm excited to take it forward with a lot of other people who are interested in this area. So, let's see. Okay, so I need to put up my disclosures. Unfortunately, Triplet Therapeutics was a company that no longer exists, but my hospital makes me declare an interest in it, regardless, because of an NIH grant that I had when it did exist. So. Okay, so Huntington's disease, let me just sort of summarize briefly, because I think most people know about Huntington's disease, but it's a neurodegenerative disease, which means it's due to the loss of brain cells or neurons. And in particular, there's an area of the brain called the striatum, which is right in the middle. Does this pointer work? Yeah, so here you can see in an HD patient brain, in this area, there's a massive hole. And so this is where the striatum is here in control people here. And within the striatum, there's a particular type of neurons called medium spiny neurons. And they're the cells that are particularly affected in Huntington's disease. And so the disease is characterized by, clinically, by abnormal movements and problems with thinking or cognition, as well as psychiatric problems like depression, irritability, or aggression. And the cause of the disease, in all cases of Huntington's disease, are caused by inheriting a change in the DNA or a mutation in a gene called huntingtin. So I put up here, this is the DNA code or the genetic code of the Huntington's disease gene. So everybody has this gene. And you can see all the letters G, A, T, C in some order here, and this is the code that makes the huntingtin protein. And what's different in patients is just this little tiny bit up here, okay? And so up here, the code is, there's a repeating letters, CAG. And the CAG gets repeated, it's called a CAG repeat mutation. So in individuals without Huntington's disease, the CAG repeat is short. It can vary between people, but it's short. And in HD patients, it's longer. It's typically 40 plus in patients. So if this CAG repeat is extended or to 40 repeats, if you inherit this length of CAG repeat mutation, then you'll go on to get Huntington's disease. So the repeat is very interesting. It has a pretty unusual property in that it's called unstable, which means it can change in length, okay? So here, so it's a dominant inherited disease, which means if you have the disease, and here's an affected individual, you have a 50-50 chance of passing that down to the next generation. And so what we can see here is an individual that has 40 repeats, and just as an example, passes this disease on to 2 offspring here. And here the repeat jumped up to 44, and here it jumped up to 45. Here is a female who passed on the gene, had 45. Here it went down, and here it went up a little bit. And it turns out that it changes quite frequently when passed from one generation to the next. It tends to go up when it's passed from males to their progeny. So this is the repeat changes, it's unstable. And so the consequence of that, let me go back one. The consequence of that is that people are born with different lengths of CAG repeats. All HD patients have different CAG repeat lengths. So that you can see here, down here on this axis here, this is the repeat length that you inherit. And each dot is a patient and shows the age of onset of that patient. So you can appreciate here that in general, the longer the CAG length that you inherit, the earlier the age of disease onset. So you can see this red line, that's the sort of average. Okay? But what you can also appreciate here, if you take any one repeat length here, so this individual 45, there's a huge range of onset ages associated with any repeat length. So that says there are other factors. CAG is really important, but there are other factors, not just the repeat length that you inherit, that can modify the age at which you get the disease. So these are modifying factors, and we'll come back to that. So I talked about the repeat changing from one generation to the other, but it turns out it does more than that. It also changes in people in the body cells over time as they age. So this is called somatic instability, or we call it somatic expansion, because the repeat tends to grow. And so what this looks like, try to represent this like this, is that when you're born, all your cells start with the same number of repeats. And so I've just, as an example, put 40 repeats up here. But over time, in cells of the body, the repeat starts to grow. It starts to expand. And the CAG repeat gets longer and longer and longer. And so you end up with some cells maybe still having 40, some cells have 45, some with 70, and you can even have like 500 or up to 1,000. And so this means that every cell has a different number of CAG repeats in the body with aging. And cells in the brain have very long repeats. So this is just to, don't worry about all the different names here, but this is just a survey of somatic expansion in HD patients across a really wide range of different brain regions. Some of these are indicated here, and other tissues of the body, peripheral tissues. And red indicates more somatic expansion, and blue indicates less somatic expansion. So you can just sort of see, these are lots of brain regions here listed, and these are lots of other regions mostly it's the brain regions that tend to have the longest CAG repeat lengths. So just to kind of make this a bit cartoony, so basically here's a brain, and each cell, if you like, has a different CAG repeat length over time. You take a snapshot, if you look at HD patient brains, each cells have different repeat lengths. So for example, here this cell has as many as 500 repeats. Here's a cell down here that has 40. Maybe this individual started with just 40, and so on. So it's like a big mosaic of different repeat lengths, cells with different repeat lengths all throughout the brain. And different brain regions have different, and different cells have different repeat lengths in them. So what we did is we asked the question, so in that we know that the disease onset can be different in patients, even if they inherit the same repeat length. So we asked the question of, well, those individuals that have an earlier onset, maybe they have more of the somatic expansion in their brains than individuals who have later onset. So we took a set of patients with early onset, and we took a set of patients with late onset, and we measured the repeat lengths in brain tissue. And what we found was that individuals with early onset, here on the left, tended to have higher repeats in their brain, more repeat expansion than individuals with earlier onset tend to have more repeat expansion in their brain, and individuals with late onset tended to have less repeat expansion in their brain. So this really provided an important clue to us that differences in disease onset might occur because individuals have different levels of CAG expansion in the brain. So we've done a lot of work in mice to try and dissect this. It's easier to look in mice because you can look over time and you can test the mechanism in mice. And so mice also have somatic expansion. And so we can focus here on the little green circle there is the striatum in the mouse. And in a young mouse with a Huntington's disease mutation in it, starts off with a repeat length of whatever you put into the mouse. And as the mouse ages, the repeat starts to grow, and you see somatic expansion in the striatum of the mouse. Okay? So we could study this in the mouse. And what we decided to study and to look at was, well, what's making the repeat expand? Why is it expanding? And what can we learn about this? And we looked at DNA repair proteins. And so DNA repair proteins are proteins of many, many different kinds of DNA repair proteins in cells, and they're required to fix mistakes in DNA sequence or damage to DNA. And so we wondered whether DNA repair proteins were involved in somatic expansion. And so what we did is you can take an HD mouse, and then you can breed it together with a mouse that doesn't have a particular DNA repair protein. So it's deficient in some DNA. And we just, we took sort of educated guesses about what we thought might be important. And so you can cross these mice together, and you can make an HD mouse that doesn't have that DNA repair protein. Okay? So it's defective in the gene that provides instructions for that DNA repair protein. So this is an example of what we saw over crossing of many different kinds of mice, knocking out different DNA repair proteins. So here's your HD mouse again, starting off with one kind of repeat length in young mice, and when this mouse ages, the repeat gets longer, somatic expansion. And so here's our HD mouse now with a defective DNA repair gene. And young mice, they start off the same, and when you age them, that repeat doesn't get longer anymore. And so we found this for a number of different DNA repair proteins. If you don't have this particular gene that makes a particular protein, the repeat does not expand. Okay? So the other thing we found is so what's the consequence of that downstream as well? So here's our HD mouse now aged with a lot of repeat expansion. And in those mice, you can see these protein aggregates or clumps. Jeff talked about. Okay, so this is actually brain slices through the striatum of the mouse, and the circles represent the nucleus of the medium spiny neurons, and within that you have these big dots, these bright dots, and these are these protein aggregates or clumps in these brain cells. And so then when you have your mouse with a defective DNA repair gene where you stop the somatic expansion, you slow that process. Okay, you either don't have it or you slow it. So you stop that protein aggregating as much. And then we asked another question. We said, okay, so this mouse has a defective DNA repair gene everywhere in every cell. It's born like that. None of its cells have this properly functioning DNA repair gene. And we know that medium spiny neurons are really important in the disease, so what happens if we just take out that DNA repair gene in medium spiny neurons? So we made a HD mouse that was defective in this DNA repair gene, but only in the medium spiny neuron. So all of its other cells have a normally functioning DNA repair gene, but its medium spiny neurons in the striatum don't. Okay? And when we did that, we also didn't see the repeat expanding. The repeat stopped expanding. And at the same time, we stopped or slowed the protein aggregation. So this told us that medium spiny neurons in the striatum have the longest repeats in that tissue, and that stopping somatic instability, stopping somatic expansion in medium spiny neurons delays the disease process. So we have done a lot of breeding of mice with different DNA repair gene deficient mice, and we wanted to scale this up so we could look at more genes and try and understand the pathway a bit better. I'll come to what the genes are in a minute. And then, so this was a scaled up strategy. This was spearheaded by Ricardo Moro-Pinto when he was a postdoc in my lab, and has his own lab now. And so to do this, what we can do is we can take our HD mouse and we can inject it with viruses, and these viruses deliver CRISPR tools. So these are tools that allow you to make specific cuts in DNA sequence and inactivate any gene you want. And it's very efficient, and we can have viruses that target the brain or viruses that target the liver. And actually, we really like to use the liver because the liver actually has a whole lot of somatic expansion as well, just like the brain. And so we can use it as a very effective readout to understand what are the different genes, and if we modify them, can change the somatic expansion. And so we can do this for one gene. And then we can also do it for many genes, targeting different genes, your favorite gene that you want, and then you can generate mice where these different genes have been targeted, and then you can look in the striatum, and you can look in the liver. So we've done a lot of work in the liver, and I'll just show you some of that, and then we're doing this also in the striatum now as well. So this summarizes some of the data. So we've looked at probably over about 60 different genes, DNA repair genes, other kind of genes. And this shows essentially above this line, anything that goes above this line, when you target that gene, you get more somatic expansion. And anything that goes below this line, when you target that gene, you get less somatic expansion. And so most things do nothing, actually. Most things do nothing. But there are some key genes that are really important, and I've just highlighted them here. And so these genes here, they're part of a specific DNA repair pathway called DNA mismatch repair. And when you knock those genes out in them, and we found for some of these we had done with those genetic crosses, and we tested it again in this system. When you knock those genes out in the mouse, you really suppress the somatic expansion. So that says that these are normally required for that process to occur. And these genes, for example, when you knock those out in the mouse, you promote somatic expansion. So their normal function is a brake on this process. So to summarize then, so some DNA repair genes are actually required for somatic expansion or somatic instability. So those are some of the genes there. And it's interesting, these genes and these proteins are normally required in cells to repair DNA. But in this context of the CAG repeat in Huntington's disease, they actually make things worse. They actually make the repeat longer. So it's sort of, when we first got these results, it wasn't what we expected. It was very interesting. And some DNA repair genes protect against somatic instability. Okay, so why do we care? So why is this important? So let me come back to this graph, which is the relationship of the age of onset and repeat length. And as you remember, for any given repeat length, there's this very large variation in onset age. And so this is due to other factors. And what are these other factors? And it's been known that this tends to run in families. So individuals that tend to have a late age of onset tend to come in family, tends to be from the same family, and individuals that have an earlier onset tend to be from other families. And so part of this is genetic, okay? And so part of it's genetic. We can try and hunt for those genetic modifiers, and we can say, what are the modifiers, what are the other genes in people that could modify, if you already have the HD mutation, but then we have a lot of other genetic variation, what are the other genes in people that could change the rate at which we get onset? And so those are the modifier genes, and so these basically contribute to having onset that is earlier or later than expected. And so over a long time now, technological developments essentially enabled you to test all genes at once. So you didn't have to pick your favorite gene, sort of like we did in the mouse. You can pick all you can survey all genes at once. And this is very important because the search for modifier genes is not biased. It's not biased by your preconceptions of what you think might be important for the disease. It's just asking, in an unbiased way, what's the genetics telling you? So what the genetics told us is summarized here. And so this is called a Manhattan plot for obvious reasons. So these big peaks, sort of like skyscrapers. And the important thing is to know is that the peaks that really stand up indicate the positions along the chromosomes of people where genes are have a that gives you the site of these modifier genes. So anything that comes up here, like these things that are indicated here, that indicates that there's a potential modifier gene. Okay, and so these, what I've highlighted in red here, so these are all the genes that have been indicated by doing this genome-wide association studies. And the ones in red are DNA repair genes, and if you remember from before, these are some of exactly the same repair genes that we know already modifies somatic instability in mice. So some of these, after these genetic results in human, we went back to the mice to test some of them. We didn't know them all before, but we knew some of them. So this was a very, very exciting finding and really told us that this really linked everything together, right? And it told us that variation in people in these DNA repair genes is altering their onset, and is telling us that this is it's altering the rate at which that repeat is expanding in somatic cells. So the rate of CAG expansion in the brain cells, is determining the timing of disease onset. So individuals who have a particular CAG repeat that they inherit, if they happen to have a complement of these DNA repair genes, a certain complement, they might have a slower onset because their repeat is expanding at a slower rate. But individuals who happen to have a different DNA repair genes, their repeat is expanding faster and they will get disease earlier. So this is the sort of simplified model. Thinking of Huntington's disease as a two-step process. After inheriting the gene, the repeat needs to expand. That CAG needs to get longer and longer until it reaches some threshold. And at that point, it elicits a toxicity process. And I put this as a big box because we don't really know exactly what that is and what the trigger is. And it may be protein aggregation, and that's a very good candidate for a process that could then lead to the cell ultimately dying. But we don't actually know exactly what those species are that is the driving toxicity. But what we do know is this needs to happen. This needs to happen first. The repeat needs to expand first. And so what this means, very encouragingly, is that if we can intervene therapeutically in this stage, we can intervene before we even get to the toxicity. So this is going very, very early. So that's what the goal is going forward. Can we find therapies that inhibit this process of somatic expansion? Okay, and as Leslie said, we just had this 2-day workshop really talking about this, which was great, really trying to sort of understand the mechanism and how we take this to therapies. So just to sort of summarize a bit of the sort of future things, so here's the disease-modifying treatment down here, right? Kind of like the light at the end of the tunnel, or maybe all roads kind of lead to Rome. Except they're not really independent roads. These all really are interconnected, but are different things that we need to do to get there. And these things are things that my lab is working on and many other labs are working on as well to try and get to. So the great thing is we have some targets now already. So we know that if we intervene, if we could inhibit some of these or enhance this one, we would be able to slow the disease. So we already have some targets, and companies are working on these. So we need to keep learning from human genetics, because human genetics got us here, got us to the gene and the modifier, so we need to keep understanding what the human genetic data are telling us, because it's the human experiment which has already been done, and we need to keep learning from that. We can try and identify novel targets, either from the human genetics or using mouse or cell models. We need to do preclinical work in mice. We need to look at readouts in patients. If we have a therapy targeting somatic expansion, we know what to measure in patients. And then other repeat diseases. Huntington's disease is one of over 70, I think, diseases caused by different repeat mutations. And this process of somatic expansion is important, known to be important for many of them. And so what we can learn from one can really help us with the other. And what we can learn from other diseases can help us with HD. And then just briefly, I talked about companies who are targeting this. These are companies who are already developing programs targeting somatic expansion. MSH3 is a key protein that these companies are going after. And finally, I'd like to acknowledge a lot of people. So these are my current lab members, many previous lab members, but I've just highlighted a few who contributed to some of the work that I showed. My collaborators, the GEM Consortium is a consortium that does the Human Genome-Wide Association study. More people than here, but I'm just highlighting a few here. And also colleagues in the field who have worked on somatic instability for a long time and have really helped us to understand this process. Funding and really thank you to the patients and their families, because we wouldn't be able to do any of this work without your support. So thank you.